Good morning, everyone. My colleague, Professor Marshall-Gradisnik, and myself are going to report on data that has recently been published in the Journal of Immunology and Immunogenetics Insights. You can download those papers from the web. Probably best if you do that because it'll give a lot more detail, colour and texture to what we're going to be talking about today. But the focus of today is in single nucleotide polymorphisms, which Sonya will talk in more detail shortly, in certain biological receptors. And the two that we have presented data on in those two publications are the transient receptor potential ion channel, the TRP receptor, and the acetylcholine receptor. Now, these receptors are part of the ancient or innate immune system, and we're describing defects that have been identified in the SNPs of those patients in our study. Now, firstly, the TRP ion channel receptors are basically threat receptors. They respond to an enormous number of threats in the environment. They're very specific also in terms of the temperatures in which they are activated by certain temperatures, and also chemicals and other elements in the environment. But these receptors are located not only on the cell surface but also within the cell. So therefore they are responding to external threats as well as to internal threats. They're distributed widely through all body tissues and all cells throughout the body. And what they do is to convert threat signals into effector activity or actions through intracellular signalling, through secondary transmitter signals in all cells in the body. And if you look at the range of threats on the upper left-hand side of the slide, they respond to osmotic pressure, volume changes, stretch, vibration, temperature, injuries, antibiotics, foreign substances, allergens, infection and irritants, and also to physical activity. And it may become obvious during the presentation that imposed physical activity on top of aberrantly functioning TRP receptors is exactly the wrong thing to be doing. And those, as I mentioned, those signals are then conveyed through second messengers within the body and then play out in terms of every organ system within the body. And starting with, and moving on to the acetylcholine receptor, these are highly distributed throughout the central nervous system, scleromuscular system, indeed the neuromuscular junction, but also remember that as part of the ancient immune system, they are distributed on immune cells in the so-called non-neuronal part of the cholinergic system. So acetylcholine is a very, very important transmitter, and also within the brain, several pathways of considerable importance there. So I'll just ask Sonya to deal with the rest of the presentation.
Thanks, Don.
I hope you can hear me. Yep, great. Okay, thank you very much, and I'll continue this presentation. The slide I'm going to present to you on this actual overhead, first and foremost outlines the cells and how cells are actually produced, or how they're coded from chromosomes are represented here. Subsequently, when you unravel the chromosome, that is the composition of the DNA, and when you actually delve a little bit deeper into the DNA, we know that the coding regions, or the bases, are those that obviously dictate the protein that's going to be produced for that particular part of the DNA, as represented here. Now, what I'd like you to focus on are these particular bases here. Changes in those bases, or a single base change known as a single nucleotide, leads to a polymorphism. And if you look over here on the right-hand side of this overhead, what you see is the gene, okay, which is represented here, has been expanded across onto this display here. What you see in this actual expansion of the gene, which was wrapped back here, is it's divided into a number of sections, those being either introns or exons. Now, in the literature, there has been a wide acceptance, probably up until the last 5 years, that the primary coding region of those genes, every gene in our body, which ultimately makes a protein, is derived from these exon sequences. However, in the last 5 years, there has been researchers that have shown that the regions here, which are called the intron regions or the promoter regions, also are important in controlling the amount of protein that is produced or showing changes in the protein function or structure. So the actual gene itself now, when you look at the functionality, it's not just dictated by the exon sequence. Rather, the intron regions are now suggested to play a vital role in the final outcome of the protein, whether it be a defect or an increased gain in function. So, the purpose of this presentation was to focus on those single nucleotide polymorphisms and predominantly examine these SNPs, as I'll refer to them here on in, focusing on these specific threat receptors, these very much primitive, innate threat receptors, these transient receptor potential ion channels, as well as the acetylcholine. The rationale for focusing on those two particular either the TRPs and the acetylcholine is they do work in unison, in part, and which I'll explain in the latter part of this presentation. But in terms of how we actually went about this study, we recruited, first of all, approximately 280 people. Of those 280 people, we screened them according to the CDC, as well as what is the most accepted additional screening mechanism is natural killer cell lysis activity. And we use that as an additional inclusion criteria. As it's widely known in the literature, that's the most consistent finding. There are a number of exclusion criteria applied, obviously in terms of thyroid disorders and others that have presented in the previous session that was here today. Sorry, my slides keep doing this. Of the 280 people, once we've applied the exclusion and inclusion criteria, 115 CFS patients were included. Now, I'll just labour on this point, and that is those 115 patients were not directly recruited just for this study. These 115 patients had been in our databases for 3 years, and we have monitored them every 6 months for the last 3 years. So subsequently, they were included in this study. Shocking at the moment for this. The non-fatigue controls, there were 90 in total, and they have also been in our database as well. A large portion of these, as in there was over 90, it was like a good portion, I think another 50. And from those we excluded them because they had started to present with other comorbidities. We then excluded obviously 75 either non-fatigued or CFS patients, and then we further then included a subject questionnaire such as the fatigue severity scale, the Kaposky performance scale, the HUDAS, which is a quality of life questionnaire, as well as the FS36. So subsequently then we undertook this study. We did it in 2 parts. Part A, looking at the SNPs in the transient receptor potential ion channels, and then Part B, looking at the acetylcholine receptors, SNPs. So the results I'm presenting here are in 2 parts. The first part is in relation to the transient receptor potential SNPs. Subsequently, it'll be followed by the acetylcholine SNP data. So this first slide here is from the first study that was published 2 weeks ago, and what we determined were there were 9 SNPs associated in the CFS group for the TRPM3. Now the significance of the TRPM3 I will outline in the latter part of this presentation. However, they were significant, and you can see the p-values here. What you see here is the allelic frequency of the CFS patients represented as frequency A versus then the allelic frequency of the non-fatigue designated as frequency U. Furthermore, when we looked at the significance for the association of CFS versus non-fatigue controls, we then further found significance in the TRPC4 and TRPA1, notably 2 in the TRPC4 and 2 significant TRPA1 changes in the CFS patients. So just to give you a summary, in terms of the 115 patients, 13 SNPs were significantly associated with the CFS patients compared to the controls or the non-fatigue controls. 9 SNPs were identified in the TRPM3. So I'd just like to labour on this point. TRPM3 with 9 SNPs, that means there's 9 single nucleotide insertions in either the regulatory regions or the intronic regions of the TRPM3. There were 2 SNPs associated with TRPA1 and 2 SNPs also associated with TRPC4. Just make a note here, TRPM3 and TRPA1, they also work with acetylcholine, and I'll show you that slide at the latter part of this lecture. So moving on now, looking at the SNPs associated in the CFS group, we reported significant associations with the SNPs of the acetylcholine muscarinic M3. And the importance of these findings are that TRPM3 and muscarinic M3 work in unison together. So this was something that was unexpected. Nonetheless, it might explain some of the symptomologies that patients exhibit, and that will be explained after these result slides. If we look here, further associations were found with the CFS group versus the non-fatigue group with the nicotinic alpha 10, 5 and 2, where the upper part of this slide designates the significant association of the CFS patients with the nicotinic alpha 10. Similarly here, the CFS patients had a significant association of these SNPs in the alpha 5 and 1 SNP associated in the CFS group with the compared to the non-fatigue group in the alpha 2. Notably, there were no significant associations with the alpha 7 acetylcholine. So in summary, those patients that we studied in part A of this investigation revealed that they had 9 significant SNPs in the muscarinic M3. And then further, when we looked at their acetylcholine SNPs, it then revealed that there were 17 SNPs associated with the same patients. Of those 17 SNPs, 9 SNPs were in the muscarinic M3. And muscarinic M3 works with TRPM3. Now, in addition, there were 5 SNPs associated with nicotinic alpha 10 and 2 and 1 SNPs associated with nicotinic alpha 5 and alpha 2 respectively. So, what does this mean? Well, hopefully this is the interesting part. So,
I bet she's got an answer.
I hope to enlighten you. So, in terms of TRPA1 or TRPA1, TRPA1 is obviously, it's been well documented, and one of the co-authors is the world leader in transient receptor potential ion channels. And he has documented nearly every single one of these key points routinely, so I'm quite proud to present this. In terms of TRPA1, its function is obviously a chemical sensor. So it's a nocisensory sensor for type C fibres in neurons. And it's well established that TRPA1 has a role in regulating neuropeptides, as well as pain sensation and inflammation. So I draw your attention to this in terms of we that is the Staines and Marshall-Gradisnik laboratory, have previously reported changes in neuropeptide receptors. Similarly, our laboratories and other laboratories have also reported changes in inflammation, changes in Th1, Th2 cytokine activity and production. In addition, if you think about the symptomology that patients also exhibit, yes, they report pain, inflammation, as well as they report migraine, headache, and neuropathic pain and muscle pain. And these are key symptomologies that could be explained by this SNP association in these patients. If we look at the TRPM3, which notably had 9 SNPs associated in the CFS patients, TRPM3 by the co-author that is Bernd Nielsen, has reported that they're located on the beta pancreatic cells. We know that there are perturbations in insulin and glucose in the regulation with CFS-ME patients. Similarly, Niels has also reported TRPM3 is located in the central nervous system. And he has, along with others, reported that these TRPM3 are associated with pain, but in the absence of actual any damage, tissue damage. So that suggests again that there's CNS impairment. Furthermore, TRPM3 is a thermoregulatory sensor. And that's a key point because the previous presenters have also shown that in the literature. If we look at what has been reported with CFS-ME patients, yes, that's a predominant feature. They have changes in their thermoregulatory functions. If we move on to TRPC4, TRPC4 is responsible for vasomotor function and smooth muscle function. It's associated with memory, attention, sensory auditory mechanisms, emotion, pain, and motor control. There are neuronal changes with memory and attention in CFS-ME patients. That's well documented. And importantly, TRPC4 also pairs with muscarinic receptors in the intestinal, gastrointestinal associated tissue or the gut. Furthermore, TRPC4 is present on a number of cells, as Professor Staines outlined, but it is well documented that they are also present on immune cells. And what is important in relation to this finding is CFS-ME patients, when they have been examined with their immune cell functions from our group as well as others, we've reported, and others as well, significant reductions in immune cell function numbers. And similarly, if we look at these actual changes, they may be mediated in part by TRPC4, in addition to the other TRP receptors that I am reporting, along with Dr. Staines. If we move on now to the acetylcholine and the predominant SNPs that were associated with this cohort, they were in the muscarinic M3. The significance of these findings are that muscarinic M3 SNPs are associated in the gastrointestinal tract. CFS-ME patients, as we know, report a number of gastrointestinal associated issues. If we look at further, muscarinic M3's receptors are also located on the pancreas, and they regulate insulin and glucagon secretion. And moreover, muscarinic M3s are also involved in the parasympathetic control of cardiac function. And we know, as well as you, that there are orthostatic intolerances associated and reported in CFS-ME patients. So, moving on, I guess looking at immune cells, because that's where our bread and butter is, looking at the acetylcholine receptors, we now know from the literature that these nicotinic and muscarinic receptors are located on T and B cell lymphocytes. And we know that they play an important role in proliferation, development, and also function. And from that provides valuable information to explain potentially the changes we see with T regulatory function, with natural killer cell lysis being reduced, with decreased neutrophil phagocytic activity, as we have previously reported in the literature. And nicotinic acetylcholine receptors are also reported in sleep arousal, fatigue, as well as those functions that are responsible for pain, memory, and cognition. So I guess in terms of the findings we are reporting here today, there are similarities with our actual SNP findings and the symptomologies that CFSME patients report. So in terms of this diagram, I guess this might wake you up if I've put you to sleep, but in terms of this diagram, I'd like to take you through this quite carefully. And that is, if you look at the top part of this diagram represented here, Professor Staines introduced you to the concept of threat, and that is threat through osmotic pressure changes, through volume stretch, through vibration, temperature, injury, allergens, and infection. We know that some CFS patients, 60%, report post-infectious viral fatigue, and then they go on to develop CFS. But what has perplexed us to this date has been the 40% that don't actually report such symptomology. But they report chemical sensitivity, such as outlined here. They report changes in cold. They report that they are intolerant to exercise or mechanical stimulation. What does this mean? If you look at this diagram here outlining the TRP, these are TRP receptors. TRP receptors are sensitive to not only, they are the responders in relation to these threats. They are also then activated, and they introduce then the intracellular signalling pathways that initiate transcription of, for example, inflammatory cytokines. If we look at the diagram here, the interaction here, TRPA1 interacts with TRPV1 here. These are two TRP receptors that are on cell membranes located throughout our body. They act together and they amplify then the inflammation that's then going to be produced. They change then the cytotoxic activity, in this instance, of a natural killer cell. They do that through their intracellular signalling and calcium mechanism outlined here on this diagram. Other additional effects are they change to elicit pain and inflammation through such as the nitric oxide synthase pathway, or NOS, and other inflammatory pathways. If we then look at the interaction then with acetylcholine, and that was the second part of this investigation, acetylcholine acts on G-coupled protein receptors. It initiates, obviously, intracellular signalling outlined here in this diagram, but it also interacts with TRPA1. And remember, TRPA1 interacts here with another TRP receptor, TRPV1. So you have an amplification in terms of pain, analgesia, inflammation. You also then inhibit cell signalling pathways. For example, reduce natural killer cell lysis, enhance Treg function, which we and others have reported previously in the literature. So, going to what Professor Staines previously reported, there are a number of threats that are perceived, and therefore their threats are acted through these transient receptor potentials, and they are amplified in terms of acetylcholine. So in summary, the SNPs indicate changes of a genetic profile of both TRP and acetylcholine receptors, and these may be upstream threats that are potentially the mechanism for the onset of CFSME. And therefore, this may then alter the function of these receptors in an aberrant way. So I'd like, on behalf of Dr. Staines and myself, to thank these funding bodies, for example, the Alison Hunter Memorial Foundation, as well as the Mason Foundation. These two funding bodies funded this exploratory work that we had a hunch with, and obviously, I'd like to acknowledge these other funding mechanisms for our ongoing research in other areas as well as these. This is the team. There are notable absences there. They're away at conferences. However, I'd like to acknowledge Mrs. Christine Hunter, wonderful woman who has been dealt a horrible blow but moved on. And I'd like to acknowledge that the team is ME, it is not CFS, and that was represented here by the lighting of our building this year, calling it Team ME at Griffith University. Thank you.
Don and Sonya, thank you very, very much for your joint presentation. A question or two, perhaps?
And it was corrected, and the bioinformaticist did that analysis, who was acknowledged on our papers at the Australian Genome Research Facility, who's the manager of the bioinformatics facility. She was employed because she's the best in Australia to do the analyses. And subsequently, the question arose, I think, comparing it to, say, a commercial database, which came up on that forum. Mm-hmm. And one would say that that's a selection bias to compare it to a commercial entity, where the bias of those SNPs that are reported are from a specific cohort of interested people to give their actual samples. Similarly, if you think about the reported SNPs, those SNPs that were reported in that commercial entity were looked at in isolation. We're not looking at these in isolation. We're looking at them in terms of the frequency of having 9 SNPs of TRPM3. And that can cause then a spliceosome mechanism to initiate a frame shift potential mutation, which then causes a change in the protein sequence, which ultimately then changes protein function.
I was just going to say, with the TRP receptors, would there be any way to model this in, for example, a transgenic rodent model, and to modify these and see what impact it has on the immune system and other parameters?
The modelling would be best utilised through an in vitro model. So, for example, an in vitro model where you isolate specific cells. Say, for example, I'll use natural killer cells as the example. Isolation of natural killer cells through the Feickel-Park centrifugation method, which is the proven methodology to utilise. Subsequently, then you would incubate those natural killer cells with a immortal cell line, such as, say, a K562 cell. But prior to that, you would label them with a monoclonal antibody for, say, for example, a TRP receptor. But you have to be careful with that. The TRP receptor is like a 7-protein transmembrane conglomeration. Yep. So when you label it, you have to be assured that you're labelling it from the outside of the cell. You can't label it cytoplasmically. It's not possible. So you could model it, to answer your question, by taking specific cells from CFS patients and then looking at those specific receptors.
Can you keep these this cell line alive for quite a long time, so you could see, longer duration changes?
You can challenge it. You can challenge it with different mitogens to then see what's being produced in terms of intracellular signalling.
Okay, all right, thank you.
Okay, thank you very much.